And I'd just like to also say what the other coordinators have said. It's been a pleasure to work with all the other projects in this cluster. I don't think I've ever experienced, going all the way back to FP5, a cluster that has worked as effectively as this. So I'm one half of the coordination team. So Rudolf would normally have been here. So Rudolf is much more the scientist, and it's his, I suppose, his brainchild, PlasticsFatE. But Rudolf is recovering from some illnesses, so he's joining online, and he will be happy to answer any questions if people have questions of him. What I will do is go through probably far too quickly, especially for the PlasticsFatE partners that are in the audience, all the results, or the sort of most of the results that we've achieved within the last 4 years. But this is really just to set the scene. So we, like all the other projects, have been looking at MNPs and the impact on human health. But what we have done is built from the bottom up. So probably we've spent more time than other projects in looking at materials and characterizing materials and developing SOPs. We've been working with the other projects in terms of sharing materials that we've produced, that other projects have produced, to get a better understanding. And what I will show in this presentation is the work that we've done in physical chemical characterization and developing SOPs, and the in vitro and in vivo models that we've used to test these MNPs, computational modeling, which is important. And I think other projects have realized that as well, because we still lack a huge amount of data, and we need to take account of the data and knowledge gaps. And then we've done some work in occupational exposure and biomonitoring. So starting from the beginning, so we, together with the other CUSP projects, developed a huge range of materials, basically an MNP catalogue. And within PlasticsFatE, we looked at, from our own sources and other sources, 21 well-defined test materials. So these cover micro and nanoplastics, different shapes, and the most common plastics that human beings could reasonably be expected to be exposed to. We developed pristine, product-derived, and aged materials for our assays. We also developed new dispersion protocols, so to make sure that these plastic particles are stable in the solutions where we're testing them in vitro and in vivo. We looked at the evolution of the corona in representative media, so that we've heard a bit about that today already. We looked at cell culture media, we looked at other synthetic media, and we looked at simulated human digestion system media, because one of the models that we looked at was a GIT. We also were involved or led 3 of the laboratory comparison studies, which I know Karina would have spoken about tomorrow, but I think Leanne will speak about that in her stead. And as with the other projects, and it will become apparent when I go through this presentation, nanoplastics have been a real issue for us to monitor, to assess. So in the latter stages of the project, we developed 2 new labeled nanopets, so one with rhodamine B and one with gold nanoparticles. We also looked at leaching of additives, and so we looked at zinc oxide, and we looked at organic stabilizers that are quite often present in plastics. What another common theme, and again, echoing what others have said, is that there is variation between plastic materials. There's variation between aged and unaged plastic materials, and there doesn't seem to be a strong correlation at the moment, certainly from the experiments that we've done. When considering fate and exposure, we looked at, and Tanya mentioned this earlier, so we looked at exposure via air, we looked at exposure via ingestion, so through food and drink, and we also looked at exposure through personal care products, and that was summarized in a paper that a number of PlasticsFatE partners contributed to a few years ago. We recognized quite early on that there were gaps in knowledge, gaps in data. So there were a number of models that were developed within the consortium to look at environmental fate, to look at occupational exposure, and to look at what happens to these particles when they get into the body. You know, how do they transfer between different tissues? What we can say is that we've taken some of the data that we generated within the project and we've put that into the models, but we're still very much at the beginning of that development. We still lack a lot of real-world data to fully validate these models. We did some early air sampling campaigns. This is basically indoor and outdoor. So indoor was in domestic properties, outdoor was in urban, suburban, and rural areas. We found low levels of micro nanoplastics in the air. Suburban had higher levels in both remote and indoor. We later in the project did some workplace sampling, and I'll talk about that when we get to that point of the study or presentation. We looked at exposure studies. So again, thinking about ingestion and personal care products, we looked at bottled water. We found minimal numbers of microplastics in bottled water. We used micro FTIR, and we used MuScan, which is a technology that one of the partners in the project had developed that's based on solid state cytometry, which has been quite good as a very quick screening of MNPs. We looked at packaged and unpackaged fruit and vegetables, and again found low numbers in these on the surface from washing. So we found that actually, bizarrely, there was more on the unpackaged fruit and veg, which we assume is from handling an atmosphere. Looking at personal care products, well, we know that MPs, microplastics, are no longer allowed to be put into the product itself. So we looked to see whether any microplastics are released from the product packaging, and we found microplastics. Now, we believe that that is coming from the pumping mechanism. So we looked at deodorants, we looked at hairsprays and things like that, and the plastics that we found most common were POM, PP, and polyurethane. So plastics that are associated with these pumping mechanisms, which are, again, fairly low levels, but these can accumulate in people's skin, can accumulate and released into the environment through showering and things like that. And in terms of FATE, we had access to medical tissues from two of the partners in the consortium. So we looked at a lot of different archive tissues. Again, I will come to the occupational studies later, and we found low levels. But what all of this work really reinforced for us is that plastics, even if you're working in a plastic-free environment, are always in the atmosphere. So we were getting lots of contamination from our study. So it's really important for the blank abstraction. Looking at FATE, we had models that mimicked gastrointestinal tract and the airway, and looking at the GIT in particular. We looked at how different systems within the GIT, different levels of GIT, influence the structure, the physical structure of the plastics that we exposed to that. And we saw that that changed with both PET and PLA. We also saw that bacteria can colonize these plastics, and that in turn, the plastics can affect the microbiomal composition of the gastrointestinal tract and the metabolism of those microbes. And comparing what we term as healthy volunteer digestion juice, or not digestion juices, but intestinal samples with those from elderly or infirm people, we see a difference. So we see that there's greater effects on the microbiome from people that are not healthy compared with those that are healthy. And we do in some models see some effects on barrier properties as a result. All of this information we developed into a QA/QC guidance document to help others with their work, and that's available through PlasticsFatE website. Then we looked at, well, what are the hazards from this exposure? Again, we had a number of in vitro models. So we had airway models, we had a large number of GIT models. Some of those were co-culture models to look at the impact of different epithelial cells with, for example, immune cells. We looked at the blood-brain barrier, we looked at the immune system, and we had a mouse model where we looked at intratracheal installation. Before I starting at, it's really important. Again, this has been mentioned by others. We spent a huge amount of effort in quality control of these materials because it was clear, we're working multidisciplinary teams that were used to just producing materials and not having any consideration regarding the sterility. And a number of the materials that we started off with were endotoxin contaminated. So we actually developed an ILS where we looked at that based on a tissue culture system to look for the presence of endotoxin contamination, and then implemented quality control to ensure that we didn't have that going forward. As with other studies, we see no significant acute toxicity, both with pristine and aged materials. Now, it's not exclusively that. So we saw that some materials, particularly the europium-doped polystyrene, did induce some effects, and PLA did in another cell line. But on the whole, there was very little. We didn't see any significant acute toxicity with most contaminated materials either, except with those that had been colonized by bacterial culture, so Vibrio and C. albicans, where we saw inflammatory and immune responses. We saw MNPs getting taken up by cells, but once they're in cells, they didn't seem to transport across cell barriers or affect barrier integrity, with the exception of the dysbiotic microbiome. And we looked at a number of different materials in this respect. So, going back to those 21 materials that we started off with, most of them went through one or more of these assays. What was clear is that the MNPs, they're getting taken up by cells. Whatever is on the MNP also gets taken up by cells, whether that's chemical or biological. And I think in the next slide, I show some of the effects of that. We also see some other effects. So in the airway model that we looked at, we saw changes in airway protein and mucus production that did vary between different types of plastics that were being studied. As I said, we see little, if any, effect in healthy gastrointestinal systems, but we do see adverse effects on dysbiotic microbiome. But with this, we see that the GIT fluids actually help reduce any toxic effects. And we didn't see any effects on the blood-brain barrier. In terms of bioaccumulation, we looked at limited food chains. So we looked in mesocosm studies. We looked at europium-labeled polystyrene uptake from soil into lettuce. I mean, IMTOX talked about basil, a similar situation. We saw lots of the polystyrene in the roots. We saw some in the leaves. Put snails on the leaves. The snails eat the leaves, but basically they crap all the polystyrene out. It doesn't go into any of the snail's tissues. In a water system, we see the polystyrene getting taken up by Daphnia. Daphnia are eaten by freshwater shrimp, and they get into the freshwater shrimp. But again, we don't see any evidence of bioaccumulation. We see overall micro nanoplastics are taken up by epithelial cells and macrophages. I mean, some of the results, which I think if you look at our poster, you'll see some pictures of macrophages stuffed full of microplastics. These are influenced by bacteria and ecocorona on the plastics. And we also looked at, well, what happens if you take a fairly common organic contaminant, in this case, benzoylpyrene, which is produced by burning plastics and burning all sorts of materials, coated that onto PET, made sure it was stable, and we saw that that was taken up into lung epithelial cell system and induced the same sort of effects that BAP would on its own, but interestingly, it also led to longer effects and effects that you don't see with BAP on its own. So the PET is actually acting as a vehicle to take the BAP into cells and induce changes in biomarker expression. Looking at the murine model, we saw lots of different lots of different nanoplastics getting taken up by macrophages in the animal's lungs. And after a 28-day exposure, where they were exposed, I think 3 times altogether over that 28-day period, we saw some translocation of these materials into the liver. We also observed that the lungs were still inflamed at 28 days, and this seemed to be most prominent for the smallest particles that we looked at, which was the PET COO2. So then we were looking, and this is obviously, it's not all in chronological order. It sounds like it's all carefully coordinated here, but we looked at occupational exposure. So to support us in that work, we had access to a clean room facility where we could mimic real production processes for plastics. So we looked at polymer melting, where we saw that there was a higher release of MNPs with PET than with PE and PP. We looked at hot blown film, and we saw no difference between aged and unaged HDPE in terms of particle release. We looked at abrasion, where PP this time had more particles released than PET and PE. And we looked at aerosolization, where we saw evidence of particle aggregation. So this was essentially to form a baseline for understanding what we would see when we looked in the real world. And in the real world, we had, I think, 10 studies altogether in different workplace environments. So we looked at waste management, we looked at packaging production, we looked at a recycling plant, we looked at a company that was making technopolymers for advanced products, and we looked at a 3D printing situation. And with all of them, we see micro nanoplastics in the air. It varies across the workplace. But what was interesting is that we saw, and it's small levels, or relatively small levels. Now, whether that would translate into health effects, we don't know. We've got some evidence for some outcomes, which I'll show in the next slide. But we're talking about a few thousand particles per cubic meter. But what was interesting was in the recycling plant, which doesn't have any active filtration going on, the levels of MNPs in the atmosphere were higher than in the packaging production plant. We looked at microbial colonization of different microplastics, and we took different aquatic ecosystems, so freshwater, river water, and saline, and we see that different bacteria can colonize different microplastics. So as others have said, this may act as a vector for pathogens. We also looked at horizontal gene transfer between microbes on these plastics, and we got evidence of that. And interestingly, we saw higher HGT with bioplastics and with tire wear plastics than with petrochemical derived ones. From the occupational studies, we looked at human biomonitoring. So we took samples of EBC, blood, urine, and looked at different metabolites, and we saw evidence of increased numbers of, or increased levels of human biomarkers. I've got, I think, 3 more slides quickly. Okay, right. So, yeah, sorry, it always takes longer than you think. We've got data gaps. So we developed models to support those data gaps. So we've got the PMCDS, which allows user to put in information. We've got an environmental, occupational, and PBK model to cover other data gaps. We combine that with other input into a web-based model that can give guidance recommendations to the user. We've supported all of this through a very heavy dissemination and exploitation engagement activity, along with other CUSP projects. We've included training workshops, stakeholder events, engagement outside the CUSP cluster on other activities, and more recently, some policy briefs which have summarized our results. And so, to summarize what I've gone through very, very quickly, pristine micro nanoplastics have few effects. They can act as carriers for chemical and biological contaminants. They can enter cells, and we see changes in gene expression. So we see upregulation of biomarkers as a result. We don't know what the long-term effects of these are, but they may aggravate underlying health conditions. So, for example, via the GIT of the elderly or ill. We recognise that we need to do a lot more in human biomonitoring, but we need to have a think about what biomarkers we're looking at. We have issues with identifying and characterising nanoplastics in real-world samples. We have undertaken ILCs together with other CUSP partners. Now that they are super important, but they're time-consuming and expensive, and we need to consider how that is done in the future. It's clear that we still lack a lot of data, so we need good models to accommodate data and knowledge gaps. And all of this data needs to be fair so that we together and others can work on that. And so, just to finish, I'd like to thank all the partners in PlasticsFatE. Have I got 10 minutes? I don't know. No. All the partners in PlasticsFatE and you for your attention. Thank you.